Okay, so we have also here the Igg antibody. So Igg antibody would have a molecular weight of 150,000. So consider here to be the smallest of all our antibodies. So with the sedimentation coefficient of 7s. Okay, there are four subclasses of our IgG. So IgG 1, 2, 3, and 4. Okay, the major functions of our IgG, so this one is the antibodies being produced on the secondary response or the secondary encounter or anamnestic response. Also provides immunity to the newborn because of the transplacental transfer. It can actually cross the placenta and therefore it provides protection for the newborn. Also capable of glutination, precipitation, and complement fixation. The same through with our IgM. Okay, for the structure, this is just a monomer, so a typical Y-shaped antibody, and therefore it would have only here, or therefore it only have your two valence or two binding site for your antigen. So that's your divalent. Okay, so we have here the difference between our IgM and our Ig antibody. So Ig antibody is monomers, so whereas your IgM is a pentamer, there are 5 subunits. Then we have also here divalent in terms of the binding site of the antigen versus your decavalent. Smallest, largest. So the IgG here is a typical Y-shaped monomer antibody. For the IgM, you can assume here a star shape if that one is on the inactive state, or crab shape if that one is active. And also, IgG can cross the placenta except your IgG3, di ba? For the IgM, that one is not capable of crossing the placenta because it's a very large na antibody. It's a protein reacting antibody. So in the blood banking, okay, we need to add potentiator to enhance the reaction of your IgG antibodies. At the same time, this one also being identified by addition of anti-human globulin. In the case of the IgM, this is saline reacting, so-called reacting antibody siya, versus your IgG, which is a warm reacting antibody, is reactive at 37 degrees Celsius. It's a clinically significant na antibody. And both of them are capable of complement fixation. Only that this is more effective because of the larger in size. We consider here the Ig antibody as co-conglutanin. So because this one is not capable of immediate clamping or agglutination action, if you have corresponding antigen, you need to enhance the agglutination action of that by addition of your potentiator or with addition of anti-human globulin in order for you to enhance the reaction. Whereas we consider here the IgM as agglutinin because this one is able to produce here immediate clamping or agglutination action if you try to combine with corresponding antigen because again the IgM is a large antibody. Okay, the next antibody type here we have your IgA. So the IgA, there are two subclasses. We have here the one found here on our serum that constitutes our IgA1 and another one found here in our secretions that become your IgA2. So these two subclasses here try to differ with one another in terms of their 22 amino acid sequence, of which 13 of that are absent in your IgA2. And because you don't have the IgA2, you do not have that 13 amino acid that render it to be resistant here against the proteolytic activity found among our enzymes found in our secretions. And therefore, they try to resist that hydrolysis of that proteolytic enzymes. And therefore, they are found in our secretion. Whereas your serum naman na IgA1, since they are not resistant to that, so they are not found in our secretions. Okay, so another thing about your secretory IgA here, the IgA2 that are found, that is found here in our secretion that contains here the secretory component. Okay, the secretory component can bind with the Fc portion of your IgA. Secretory component is a 5 immunoglobulin-like structure produced by our epithelial tissue. So once it's tried to combine with your IgA, so the secretory component try to perform the following functions. This is responsible actually in preventing the hydrolysis of your IgA in your secretions. and also allows the transfer or the transport of your IgA among the surfaces. Okay, so we have here the structure. So secretory IgA is a dimer, so it contains a J chain. So therefore, it would have also four na antigen binding site, becoming that one as your pentavalent. Your serum IgA, on the other hand, is a monomer. That one would have only have divalent lang, two antigen binding site. Okay, the next one, we have here your IgE. Other name for that is your reagent. So this is the antibody that is being produced here in response with our allergen. So this involved here in our allergic reaction or even in your parasitic infection. So IgE considered that one as the least abundant among our immunoglobulin, constituting only approximately 0.002%. So bakit konti lang siya sa ating circulation? Because majority of the IgE has a receptor. Meron siyang FCRI, FCERI receptor. Meron siyang receptor with your basophil and our mast cells, and therefore most likely they are found bound. Nakakabit siya kay basophil and mast cells, so therefore they are not found in our periphery or not found in our plasma. And once they are triggered, they try to be responsible for the manifestation of your allergic reaction. Okay, with those signs and symptoms of your allergic reaction. Okay, for the structure of your IgE, this one is monomer, so it will have the divalent antigen binding site, but it would have an extra constant domain. So up to CH4 siya, katulad ng IgM natin. Okay, so we have here the IgD. So the IgD, on the other hand, so this is the most susceptible here to your proteolysis. It's because of extended, may mahaba siyang hinge region that become the target of your proteolytic enzyme. So madali siyang masira because of that. Okay, so this one serve only as a receptor for our B cell along with your IgM. Actually, the IgD try to serve here or help your B cell in order for that to have a contact with the antigen, to bind with the antigen. It also determine the lifespan of your B lymphocytes. Okay, for the structure, that one here is a typical Y-shaped antibodies monomer, so it would have only have, okay, a divalent antigen binding site. Okay, so we have here the type of our immune response. We can divide that one into your primary and secondary immune response. So when we speak about the primary immune response, is refer here to the first encounter with your antigen. Okay, so the end result of the primary immune response would be the production of your memory cell. Okay, and your secondary or your anamnestic immune response refer here to the second exposure for the same antigen. And that can be recognized already here because of the development of your memory cell that's being produced during the primary immune response. So we have here the stages for our antibody production as when our body has been exposed to a foreign antigen. So divide here into four stages or phases. So we have here the first one, we have the lag phase. Okay, the lag phase here is just the orientation of your antigen. So the antigen enter your body and it would allow the simulation of your immune response. But this is the stage wherein you are not yet producing an antibody. So you have your natural immunity which try to identify your pathogen or antigen for antigenic stimulation. And try to enter here the lag phase, the LOG phase, characterized by an intense antibody production. Since dito magkakilala na sila, and eventually there's already a phagocytosis, antigen presentation, it will stimulate already the antibody production. And with it, when it's already here on the next stage, what you have for that would be an intense antibody production. Dadami na ng dadami ang antibody natin. That's your logarithmic phase. Steady state or the plateau state here would eventually be characterized by the balance between your antibody production and antibody degradation or death. Ganon. And the decline phase or the disappearance phase characterized by an increase in antibody degradation compared with your antibody production. So if you are asked when are you going to When is the best time to perform here serological test in order for you to detect the presence of the antibody? So, dapat dito tayo sa steady state. Although in your lag phase, LOG phase, you have already antibody production, but you need to use here a more sensitive test in order for you to detect the presence of your antibody. Pag dito ba lang na phase, so wala ka madidetect na antibody. And dito naman, medyo pababa na siya, it might also have the possibility of having the false negative result for that. Okay, we have here the difference between our primary and secondary immune response. So again, the primary immune response would have a primary immune response would have a longer lag phase because it's just the first encounter. So medyo matatagalan sila ng getting to know each other. Comparing with your secondary immune response would have a shorter na lag phase because again, as a product of your primary immune response or primary encounter of the antigen, your body is already developing a memory cell. It's being programmed primarily to remember that antigen, which has been already in the past, which has responsible for the stimulation of the antibody production, and nakilala niya na. And therefore, it would have here a shorter lag phase. Then it try also to eventually go through with other stages. Okay, the predominating type of your antibody in the primary immune response would be your IgM. And if you have already your secondary immune response, it's capable of class switching. It can switch from the IgM to IgG and become an IgG, signifying that one that's your secondary or your past infection, ganon. We have also here antibody affinity and antibody titer. So we have here primary immune response, we have decreased compared with here with the secondary, so we have a much higher affinity for the antibody. Okay, we have here some terms regarding our antigen-antibody interaction. So we have specificity. So it's just the binding of your antigen with specific antibody. So that's how specific it is because again, your antibody is being produced specifically for a specific antigen that try to stimulate its production. It will only react on that on the basis of their paratope and the epitope. Cross-reactivity, however, so those are some antibody wherein they can cross-react. It can react with other antigen, although that antigen is not the one causing its production, provided that antigens share the same or some homology or resemblance with the antibody or the antigen which responsible for stimulating the production of that antibody in the first place. Multivalent antigen naman, so this refer to your antigen capable binding with multiple antibody. So one antigen, madaming antibody, multivalent. Pag cross-reactivity naman, isang antibody, madaming antigen, kaya niya mag-react to your cross-reactivity. We have here the term affinity. So affinity refers here to the force of interaction or attraction between the epitope of your antigen and the paratope, which is found on the Fab. Makikita natin ang paratope, which is the antibody binding site of your antibody in the Fab region or Fab fragment of your antibody. Avidity naman, so ito pag sinabing affinity, so na-attract sila sa isa't isa. Pag sinabi namang avidity, refers here to the strength of the force of interaction between your antigen-antibody complex. Kung ito affinity, magbabind pa lang siya, the force na magbabind, that's your affinity, hindi pa nagbind, that's your affinity. Pero pag avidity, nagbind na silang dalawa. We're able to form an immune complex primarily made up of your antigen and antibody, and the total sum of their complex, the force of that's your avidity. Okay, so we have also here the area in our antigen-antibody reaction. So we have here the zone of the equivalence. The zone of the equivalence is the area on which your antigen concentration is equal with your antibody concentration. This is the area, ideally, on which your agglutination reaction we're going to take place. So, meaning to say your agglutination reaction we're going to take place if you have an adequate concentration of your antigen versus your antibody. We have also here the area prozone. Okay, the prozone here is the zone of your antibody excess. So you have much antibody compared with your antigen. Okay, and we have also here the post zone. The post zone here would have more antigen compared with your antibody. Both this area, pro zone and the post zone, since they are not of equal concentration between your antigen antibody concentration as your reactant, it would result here to your false negative reaction. Wala kang reaction. In this case, in the pro zone, since you have more of your antibody compared with your antigen as your agent, all you need to do here is just need to dilute your sample because the sample contains your antibody para magkaroon ng adequate concentration between your antibody, your sample, and your antigen as your agent. In the case of the post zones, since antigen exists ito, wala kang choice. But eventually, to just wait kung kailan magpuproduce ng antibody ang ating patient. Okay, so or until nag-produce siya ng, magdadami ang kanyang antigen, So, what you're going to do with that, parang early, it would have a early detection. So, for that, you can recommend that the patient will be having a repeated examination on next week, or the following week, or following day, para mag-increase ng kanyang titer ng kanyang antigen. Okay, we have here the detection of your antigen-antibody reaction. We have a different test procedure that can help us then to detect your antigen-antibody reaction. You can use here the agglutination reaction. So agglutination reaction is just the reaction between your antibody plus your particulate antigen. Pag sinabi naman natin precipitation is just the reaction between your antibody plus your soluble antigen. So particulate, soluble, this the term utilized here for your agglutination and your precipitation. You can also detect your antigen-antibody reaction through your hemolysis here, or you can also your heme agglutination reaction. You can have your RBC as the carrier of your antigen. So, nasa RBC na surface ang antigen natin. Okay, if you wanted to detect, for example, ang antigen na nasa RBC na patient natin, so lalagyan mo siya ng antibody as your reagent. If the sample of the patient ay may corresponding na antigen with this antibody, it try to bind your antibody on this one and it would try to cause lysis of your RBC. Kaya this become here your hemolysis and consider here as positive reaction. We can also identify our antigen-antibody reaction here with your labeled immunoassay. We'll be discussing this one. Sa may topic tayo ng labeled immunoassay. On this one, so ito siya, like, if you are detecting antibody on the patient sample, mag-a-add ka ng antigen as your reagent. Plus, mag-a-add ka din ng another reagent, like, for example, anti-human globulin labeled with the enzyme. May naka-attach dito na enzyme. So, like, for example, if your patient sample has antibody, nag-add ka ng reagent na antigen, magre-react siya dito. Mag-form ka ng antigen-antibody complex. Pag nag-add ka ng panibagong reagent, which is anti-human globulin labeled with the enzyme, magbabind siya dito sa antigen-antibody complex na na-form mo because your sample has antibody. And what you're going to measure here would be the activity of this enzyme. Okay, so like, depending on kung ano ba siya competitive or non-competitive na immunoassay. So, that can also detect here your antigen-antibody reaction. Okay, we have here the terms for the variation of our antibody. So we have here your isotype. Isotype here would be the variation of your antibody in terms of the heavy chain. We have also here the allotype. So that's the variation of your antibody in terms of their constant region. And we have also your idiotype. It's the variation of your antibody based on the variable region. Okay, then we have here the genes coding for the immunoglobulin antibody or the antibody production. So again, as a review, our antibody is made up of the light chain and the heavy chain. Okay, so first we're going to make here your heavy chain. So itong heavy chain natin. Diba? Your heavy chain, diba, as what we've discussed, made up of the variable region and we have also here the constant, okay, na region. Okay, our heavy chain is encoded in chromosome number 14. So within the chromosome number 14, there are genes coding here for the variable region of your heavy chain. In the variable region of the heavy chain, so itong part na to, there are 3 genes for that, your VDJ na gene. And again, the constant region here, okay, can be your mu, delta, gamma, alpha, or your epsilon. So whatever kung ano ang mag-bind dito, like for example, okay, so VDJ because of the constant, okay, so automatic na ito na meron kayo isang VD saka G na gene, VD saka J na gene, then 1. Mamimili ka lang dito ng ating constant. Like for example, if you have your VDJ plus mu, that become your IgM. So mu heavy chain ang maproproduce mo dito that eventually become your IgM. Okay, now for the light chain naman, so your light chain, so itong light chain natin, so the light chain is also divided into your variable region and we have also your constant region. Okay, the variable region here of your light chain would contain this gene, VJ na gene lang. And the constant region could be a kappa or lambda. Mamimili lang siya doon. Okay, for the kappa, okay, that one, so kappa here, dito yung constant niya. So itong part na to, mamimili siya either your kappa or your lambda. Okay, your kappa, gene here encoded in chromosome number 2 and the lambda is encoded in chromosome number 22. So mamimili siya, VJ, kung magre-recombine dito sa ating kappa, kung successful siya with that, so that become your kappa na light chain. Pag hindi siya successful, ito pipiliin niya. But never both ang kanyang light chain. It could only be either a kappa or a lambda. Okay, so thank you.